So I'd like to tell you about some work we've been doing in the Redwood Center for Theoretical Neuroscience on trying to find patterns of activity that are embedded in high-dimensional data sets. So first of all, by way of introduction, the Redwood Center is basically a group of faculty, postdocs, and graduate students. We're over in Evans Hall, and so basically a lot of us have backgrounds in mathematics and computation and physics, and we're trying to bring ideas from these fields to bear on neuroscience to try to understand the principles of information processing in the brain. So I'll use this slide by way of just introducing the people whose work I'm going to be highlighting today. So one is a student in my lab, Amir Khosrowshahi, who's been fitting some of these models to data from cat visual cortex and monkey visual cortex. And then also Charles Cadieux and Kellyanne Kupsel, who have been developing these multivariate phase models and fitting those to a variety of large-scale neuronal recordings. So the problem we're facing in this field, in general, in neuroscience, is that we're trying to understand the principles of operation of a system that's composed of many elements, which are all very tiny and highly interconnected and interact with each other. And so, of course, the number we always hear in the human brain, there's approximately 10 billion neurons in the system, and so that's just sort of a very difficult number to get a handle on. And this slide from David Hubel, this picture from David Hubel, sort of illustrates kind of the almost futileness of the situation we face of the investigator putting a single microelectrode in the cortex and recording from a single neuron out of this collection of 10 billion neurons in the system. And the staggering sort of realization of this, I think for me, is that most everything we know in neuroscience today, not only in terms of just the raw data, but in terms of our conceptual understanding of the brain, has been built up from a process of recording from neurons one at a time, okay, without being able to observe the interactions among these neurons. So we've learned a humongous amount just by doing this, recording from one neuron at a time. But still, I think, and many of us in the field believe now, we're missing a large part of the picture because we have not, until very recently, been able to get a handle on the interactions in this system. So what's changing this landscape really dramatically and very quickly over the past decade is the development of a number of new technologies that are being quickly adopted in many labs now in neuroscience. So one is the so-called Utah array, which is a kind of silicon probe technology of basically sort of a bed of nails that are put in the cortex and you record simultaneously from many neurons. Another, the so-called silicon polytrodes, which Tim Blanche here has been instrumental in pioneering. Basically, it's a shaft of silicon in which the electrode contacts are made all into one piece of silicon here, and that allows you to look at, monitor the activity simultaneously across the many layers of the cortex. The so-called ECOG arrays, which Bob Knight's lab has been instrumental in pioneering and using here, which has been used a lot in human cortex, and then also 2-photon calcium imaging data, which allows one to monitor activity of a three-dimensional volume of tissue, albeit on a slower time scale, because it's not direct electrical activity. Okay, so all of these new technologies are presenting us, it's very exciting. It's allowing us to see things that we haven't been able to see before, but it's also presenting us with a very difficult and challenging question, which is how to make sense of it. And it's kind of interesting, I think, if you went back in the field 10 years ago, at least this is my personal experience, and I don't know if there are people still around like this, but many of my colleagues in neuroscience, I would ask them, well, why don't you try to record from multiple neurons simultaneously? And the answer I got from them is, well, why would you ever want to do that? Or what would I do with that data if I had it, right? They were just sort of mystified. It's like, well, I don't know how that would help me other than just increasing my yield. And I think part of the reason for that sort of perplexedness in the answer is that it's really it's difficult for all of us to answer that question. What do you want to look for in the data? And so one obvious question is you'd like to be able to look at circuit interactions, but of course you don't have access to the entire circuit, only to a fraction of the circuit. So you're not going to be able to learn everything about the circuit. So what I want to tell you about today is some work we've been doing in the Redwood Center, just focusing on two different methodologies that have come out of our group there. One based on trying to fit these sparse latent variable models, and another based on trying to fit these multivariate phase distributions to data. So the first part I'll focus on this work by Amir on fitting sparse latent variable models, and this is to data from Charlie Gray's lab at Montana State University, Bozeman, from cat visual cortex and also macaque monkey visual cortex. So the idea of the model is shown here. This model, this sparse latent variable model, actually came out of computational neuroscience, and initially out of attempts to try to understand the structure of sensory data that's coming into the system, to model the structure of natural scenes, of natural sounds, and so forth. And so the way this model has been applied previously to capture the structure of natural images is just shown here. If you have image patch disk intensity as a function of space, then we're thinking about sort of a little patch of cortical neurons analyzing a little patch of input image data. Then we're trying to think about how that image data could be described in terms of a set of features, which are these basis functions phi sub I. And generally what we want to do is learn these features. We don't know a priori what's the best feature set to describe natural scenes, because these images that come in have a very complex structure to them. Okay, so the idea is we want to learn that structure. What's a good structured basis in which to represent this data? And so we can do that by imposing sparseness on these latent variables a sub I, which basically weights how much of each of these features is present in the image. Okay, so sparsity is imposed in this model by putting a penalty on these coefficients a sub I, which penalizes them for being active. And by doing that, by then fitting, you can fit this in the context of a probabilistic model to the data just by maximizing the log likelihood of the model. And then that gives you a way of learning the features that are best adapted to the data, to the statistics of the data in terms of this kind of model. Okay, so the idea of sparsity here is really just sort of expresses our desire to come up with a parsimonious or simple description of the data. The extreme, if you push sparsity to extreme, you're doing something like vector quantization or clustering, right? You would just be describing the image data in terms of one feature at a time. What's the best sort of feature vector that describes what's going on? Instead, sparsity is backing off that. It's saying, well, I don't necessarily want to have one at a time. I could have k at a time, where k is small. Okay? And I'm going to allow these things to superimpose. So I end up getting a sparse distributed code of the data. Okay? So I'm not saying I'm forcing it to be described in terms of one feature at a time, multiple features at a time, but I want to keep that number small. Okay? So that's a way of forcing the system to discover the structure contained in the data. Okay, so this model has been applied to image data. When you do so, it gives a very nice result, and it reveals these sort of edge-like patterns. If you initialize these basis functions to random initial conditions, it pulls out these sort of edge-like functions in a sort of multi-scale dictionary, very reminiscent of wavelets, but also more importantly, similar to the response properties, the receptive fields, measured receptive fields of cortical neurons. More importantly, though, this dictionary that you get out of adapting a sparse code to data is very different than what you would get out of another kind of linear model, principal components analysis. Okay, this is a standard linear model that's been used, that was popularized in the social sciences, and but the problem with PCA is it only learns from the pairwise statistics. It's basically fitting a Gaussian model of the data. And all the interesting structure in natural scenes and many forms of data resides in the higher order statistics. Things like the fact that there's oriented edges in the scene and so forth cannot be captured by a Gaussian model. And that's revealed here. When you fit PCA, you don't get anything like edges. It doesn't tell you. It's basically blind to those forms of higher order structure contained in the data. Okay, so when you apply this to sparse coding method to natural scenes, it pulls out that higher order structure and gives you complete code of that data. As well, we can apply this to movies. We can similarly extend that through a convolution model where these basis functions now have a temporal component, and they're convolved over time with a set of sparse latent variables. Okay, so now we can learn a set of space-time functions which describe the structure in movies. And again, you get a kind of very intuitive result which says that movies are basically composed of a collection of moving edges. Okay, so you can come up with a very sparse and compact description of a little sort of section of a movie in terms of how edges are moving at any given time in the scene. And again, this is a complete code that completely tiles the space. You can describe any movie just by superimposing these functions together properly. Okay, so that's applied to image data. These are previous results from long ago. And the idea here is we want to do the very same thing now on all this multi-unit activity we're getting from the cortex. Basically take all the data that you get out of sticking a multi-unit electrode in the brain, treat that data like a natural image, and say, what structure do I find in there, just in the same way that we asked with natural scenes. Okay, so this is the back of a cat. Well, yeah, no, I don't think cat, monkey, I think, in this case. Whoops, wrong animal. Okay, so anyway, so the idea is just to sort of zoom in on it. Basically, what I'm going to show you here is there's a cross-section. The point of that is just to show that what we're showing is a cross-section here, a cross-section of the cerebral cortex. And just to sort of show you the geometry of what's going on in these recordings in Charlie Gray's lab, he's using a certain kind of silicon polytrode, which has 32 channels. And that 32-channel polytrode is basically inserted perpendicularly to the surface of the cortex. And that's basically approximately the location of the recording in this case. And this is just showing a Nissl stain of the cortex, which is basically folding around at this point. Okay? And in the Nissl stain, each one of these dark spots, if you zoomed in on them, those are individual neurons, okay, that are being filled there. Okay? Just to give you an idea of the scale. All right, so basically what we get out of this now is kind of like a 32-channel movie, right? And you can look at this data. If you just looked at the raw data, it would sort of contain, it would be very broadband, contain large-scale fluctuations. So usually people look at this data filtered in different frequency bands. In this case, we're looking at the high-pass filtered data in the range from 500 to 10,000 hertz. And this is just showing you kind of a movie that Amir generated from this data of all the recordings coming from all 32 channels simultaneously as a function of time. This time scale bar is at 25 milliseconds. The whole thing here, I believe, is 250 milliseconds. Okay? So this just gives you an idea of the kind of data that you're getting out. And all these little blips here are basically neurons spiking, right? So the simultaneous activity of neurons at different depths, right? So this is the superficial layers at the top, and this is the deep layers at the bottom. And we're getting kind of a simultaneous picture of all the activity going on here. If we zoom in on the time scale a bit to magnify that, now you can sort of see the shapes of the individual waveforms. So this is presumably an individual spike here, another spike from the same neuron, different neuron up here. So any given neuron shows up with a certain temporal signature that's mixed locally onto a small number of electrodes. Okay? And obviously then neurons that are proximal or close to each other will have different temporal signatures, but they'll tend to overlap spatially. Okay? So one problem we face here is a kind of demixing problem. So just kind of looking at all this data, going in and finding what are the individual unitary events of single neurons that are creating these observed waveforms here. Okay? And so that's the Highpass. That's the Highpass time scale. And the low pass, which is typically referred to as the LFP, or the local field potential, you can make a similar kind of movie, except now the time base is much larger. This whole time is 5 seconds here. The scale bar is 500 milliseconds. And again, so you can see these kinds of, this is very effective at revealing the large-scale oscillations in the system, how ensembles of activity tend to be oscillating together. And you can see that this activity is sort of localized to certain layers, has certain frequency bands that it's occurring in. And again, we want a way of kind of going in automatically and finding out and pulling out what are the sort of activity patterns that are best suited to describe the space-time data. Okay, so the idea again that is to apply this sparse latent variable model. So to now instead of image pixels, these are recorded waveforms. So I indexes which channel you're recording from, and X is the voltage as a function of time. And we're going to describe that now. We want to describe the raw data in terms of a set of learned features. Okay? And so we don't know what these are a priori, but we want to learn them and adapt them to the data so as to describe the observed data in terms of a set of sparse latent variables, coefficients that are as inactive as possible at any given point in time. So at any given point in time, I should be able to describe what's going on in terms of a small number of these features here. Okay? And we do that again the same way as before by imposing a penalty on becoming active here. And we could formulate this again in terms of a probabilistic model and trying to maximize, learning these features by maximizing the log likelihood of the model. Okay, so when Amir fit this model to, first of all, the Highpass filter data, which contains these, really is the best regime to look at the spike waveforms of individual neurons. These are the learned basis functions you get. So what was starting out there when it initially started, that's just showing you the initial condition is basically random, because we're not assuming anything a priori about the structure of these things. So they start out completely random. Get it going again. So these functions start out completely random, and then they quickly converge to a solution here. And this is not in real time, but elapsed by, I forget what scale factor, but it's basically sped up here. And each one of these boxes is showing you a different learned basis function, phi I. So I'm sorry, phi j. So I indexes which electrode is trying to be explained. Okay, so the 32 different channels in each of these functions here, from top to bottom, and the time base on each of these basis functions is 2 milliseconds. So each of these spatiotemporal functions is 2 milliseconds long. Okay, so what you're seeing here, so that just started over again, I'm just repeating over and over. What you're seeing here is, for example, many of these functions are converging on single units. So Amir has been able to show that many of these learned functions resemble the spatiotemporal signatures of actual neurons that have been spike sorted using other more conventional methods. Okay, so what we're hopeful of is that we might have a method here of automatically pulling out these spatiotemporal waveforms from the data in a more parsimonious way that allows, especially for superpositions of these patterns, which basically clustering methods are not going to handle very well. Another thing that this method does is it separates artifacts from signals. So these other kind of weird, ugly functions that we see here are sort of signal artifacts that are sort of spread across all the channels of the electrode. And so it nicely isolates those artifacts onto a different set of basis functions. And so potentially provides a way of denoising the data or removing artifacts by simply subtracting these components out of the data once they're fit. Okay? So this is something we're kind of excited about. And another neat thing about the way that Amir has been able to apply this method is that it can be done, again, not in real time, but almost close to real time, that during the recording, as they're putting the electrode into the cortex, they can actually run this algorithm, pull out these basis functions during the experiment to get an idea of where are the neurons along the electrode, so they know how much further they should go, or if they should pull back and so forth to get the electrode in a good position, so that they know they're getting isolated spikes across the length of the electrode. Okay, so it's a very efficient method that Amir has been able to get going here. And then, so that's on the high-pass filter data, and we similarly applied it to the low-pass filter data. This is from a different polytrope with 54 channels, using a somewhat different method for fitting the model. So again, since it's low-pass filter, the time base is much larger. So each one of these learned basis functions now has a temporal extent of 250 milliseconds. And the thing that's interesting here is what's being pulled out by many of these functions is oscillatory patterns. So, for example, this I think this pattern here and these ones here are oscillations in the gamma range, and some of them extend across all the layers of cortex. Others of the oscillations, for example, at a different frequency band here, are confined to just the superficial layers, and others to other patterns just to the deeper layers and so forth. So again, what this seems to be doing is pulling out patterns in the LFP contained in the data that have specific frequencies and specific laminar distributions to them. Okay, and so potentially what one might be able to do with these recovered components is use these as regressors in a model to try to find out what their functional significance is. Okay, with relation to certain features in the stimulus, and so it might be more informative to use these components as opposed to the raw data. So, and another way these have been applied is to it's the 32 channel array that Charlie's been getting going in the lab. And this is a macaque monkey. So with this 32-channel array, it's just sort of an array of electrodes here that's spanning across 3 different visual cortical areas, V1 here, V2, and V4. So what this allows us to do is simultaneously look at the activity in 3 different visual cortical areas. And so Amir's done a similar thing here of learning basis functions, spatiotemporal basis functions, to fit that data. This is the full set of basis functions learned. Of course, they're too small, you can't see them. But zooming in on just 2 of them here, we can see some interesting patterns. So this is just showing you this, first of all, the segregation. The red waveforms are those sitting over V1, the green over V2, and the blue over V4. And in this particular basis function has pulled out a component that seems to pick up on synchronous activity between V1 and V2. These sort of synchronous spikes here. This particular component seems to be pick up on oscillations, gamma band oscillations that are sitting in V4, but not so much in V1 and maybe a little bit in V2. Okay. So again, this is these are just sort of early days and maybe a sneak preview of what's sort of being found, but it's something where we have high hopes for and be able to get a better picture of what's actually going on in this data. Okay, so a second method that's been developed in the group is and this is work by Charles Cadieux and Kilian Kopsel, is fitting these multivariate phase distributions to data, and this method is particularly well suited to describe the structure of oscillations recorded across different multiple recording sites in the brain. So the first comment here, and this is kind of something that's remarkable in neuroscience, is that in many parts of the brain and in many different animals, neurons oscillate. Why they oscillate, no one really has any idea. I think theoretically, it's in terms of why. Well, people have ideas, but nothing's been proven about why it's doing this. But it's an interesting phenomenon that's been the subject of a lot of controversy and skepticism over the years. And I think there's been renewed interest in these oscillations over the past 5 years, especially, in understanding them. And they're being revealed in many more systems now. These are just 2 examples in locus olfactory system and cat V1. Okay, so the problem one is confronted with if one wants to, if one is now recording from multiple areas of the brain, and this is data from Jose Carmena's lab that I'm going to show you. So, but I guess this is a human brain here. So the problem one is confronted with is that if you're recording the LFP from multiple sites here, and let's say single unit activity from other sites, we want to somehow take the oscillations that are observed in multiple sites here and describe the relationships between those oscillations somehow, right? And especially describe the relationships between those groups, that group structure in the oscillation, to single unit activity recorded at specific places in the brain. Okay, so what they've developed basically, what Kilian and Charles have developed, is a way of fitting a model to describe the phase coupling among a network of neurons. And what they've done in this work, which Ryan Conolti was the lead author on, is to show how a model that's fit to an ensemble like this can actually be used to predict the activity of a single neuron very effectively, just in terms of the phase coupling of the network. So I want to tell you a little bit about more about that in detail. Okay, so to appreciate the problem here, whoops. Okay, so it's first of all, I think important to understand what is the difference between fitting a model of the phase coupling versus simply measuring phase correlations between recording sites, which has been pretty much the prevalent method up to now. Okay, so this is just showing a picture. Let's say you have 4 recording sites, and you see oscillations going on in each of these 4 sites. And let's say these are the true couplings. This is the true network structure of those 4 recording sites. Okay, if you simply measured the phase correlations, right? If you just measured the pairwise phase correlations in these 4 recording sites, you would see, in this particular example, a strong coupling here. You would actually see no coupling here, no phase correlation here, because it ends up getting canceled out by other interactions in the network, okay, and so forth. So if you just measured the pairwise phase correlations, you would get a very misleading picture about the network connectivity. It's only by fitting a model of these phase of the phase correlations that you can try to recover the actual estimated couplings. This problem is well known in the social sciences. Again, this is basically what factor analysis does, right? When you try to, if you take a bunch of variables that you're measuring, you can measure the pairwise correlations between all those variables. Those pairwise correlations do not tell you the actual causal model, the actual causal structure of that data. You have to fit a model. That's what factor analysis does. Or if you're fitting a Gaussian distribution, you're basically taking the inverse of that matrix, which is the inverse of the covariance matrix. And it's the inverse of the covariance matrix which tells you the couplings between the elements in the data. Similarly here, what we want to do is fit something like a multivariate Gaussian distribution to this data. The problem is that the variables that are being fit here are circular variables. They're phase variables, okay? They're telling you which phase of the oscillation the activity is in, not a real number. Okay, so you can't fit a multivariate Gaussian distribution to circular variables such as phase. Okay, you can, however, write down mathematically a model distribution that has a similar form to a Gaussian, right? Which is determined by these pairwise couplings in this matrix K. Okay, but fitting this matrix K is not as simple as measuring the pairwise correlations. If this were a Gaussian, then K would simply be the inverse covariance matrix. Okay, but we can't. But the fitting procedure is not as straightforward. So what Charles Cadieux and Kilian Kupsel developed was basically a method based on score matching to fit this model to the data, which is very efficient and which you can converge upon a solution for K given the pairwise correlations in a very short amount of time. Okay, so that's essentially. This is essentially basically the mathematics and the computational development that allows this method to work now, that Ryan Canolti used to fit the data from Jose Carmena's lab. So what they did here then is they tried to use a model of the phase coupling among a population of neurons to predict individual spike activity. Okay, so you can write that down very simply as the probability of getting a spike from an individual neuron. Okay, if you're recording from an individual neuron, what's the probability of getting a spike from that neuron given the observed phases of a bunch of other sites in the cortex? Okay, those may be neurons right nearby, or those may be neurons very far away. Okay, but given that you know the phase of the oscillation that those other neurons or recording sites are in, what's the probability of getting a spike now from this particular neuron? Okay, that's what we want to do. And Bayes' rule tells us that in order to compute that probability, we have to know two things. Well, the prior probability of a spike, that's easy, but we have to have a model for the probability of a given set of phases arising when you observe a spike. Okay, so this you can measure. You can measure, again, the phase correlations, but in order to fit the distribution, you have to get that coupling matrix K. Okay, and then that's going to be weighed against the average phase correlation model here. So when you sort of flesh that out in terms of a model, in terms of all the math, what you need is essentially the ratio of these two terms, P of theta given spike versus P of theta. And that's sort of given very elegantly just by the difference of these two coupling matrices. Okay, so they fit these coupling matrices K to the data, and that's what allows them then to have a predictive model of when a neuron is going to spike, given the observed phases of other recording sites in the network. Okay, so this is just showing the results they get when they fit this model to the LFP and single unit data from the Carmena lab. This is three particular areas. They had an array of neurons sitting over M1 motor cortex in the right hemisphere, over M1 in the left hemisphere, and over an array of neurons in the premotor cortex, okay, in the left hemisphere. And these dark lines show basically those coupling terms K in that matrix that you fit to the data. And once you fit this coupling matrix, and this is for basically the best predictor for a particular neuron, you can show that it generates a very nice prediction of activity. So this is just showing you the correlation between the predicted activity given a set of observed phases across these recording sites versus the actual measured firing rate from a particular neuron. And you can see that you get a really nice fit. I mean, you know, in terms of this trying to fit neural activity in the cortex, this is about as good as it gets anywhere. So it's a very, so I think they've discovered a really effective model of being able to predict the activity of a single neuron given the global state of many other recording sites in the brain. And mind you, they're not looking at, you know, what stimulus is being delivered or even what the motor function is of the system or anything. They're just looking at other recording sites in the brain. And this is a really kind of remarkable thing that the ability to predict the neuron depends not so much, the neuron's activity, it depends not so much on the proximity of the neurons, but basically what network these neurons tend to be coupled to. So this is just showing the predictions of two different neurons in very different parts of the brain, one in the left hemisphere and the other in the right hemisphere. Okay? And they have very similar predictions because they have very similar networks that they're connected to using this fitting procedure. And these are two neurons that are very close to each other, in fact recorded off the same electrode, yet they have very different predicted activities because they appear to be connected to very distinct networks in the brain. Okay, so just because you're next door to someone doesn't mean that you're going to have similar activity or be even coupled to the same network. Okay, and that could only be revealed, I think, by a method that was able to sort of go in and capture this sort of these patterns that are embedded in this high-dimensional data. Okay, so that's basically summarizing what I just told you, that these new technologies are presenting us with new challenges. We think we've developed some algorithms that meet these challenges, or at least give us initial hope that we're going to be able to find some interesting and new patterns there in the data. Okay, so thanks very much.
Thank you so much. So we have two quick questions. Fascinating talk, very interesting indeed. One quick question is, you know, if you think of something like a time series, and you think of association versus causality, right? I mean, these are still based on association. So strong association seems to make excellent sense. But is there any particular requirement to verify causality or not necessarily?
Causality, you mean of one part of the network on another?
Yeah, that's right. Because you think of the extension is you're doing it in time series form, but if you think of simple regression is an association or correlation, but it's not causality. And so you need something else to establish causality. So, but right. Here, I mean, the evidence seems pretty clear just because it seems to make common sense, if you will. But I'm just curious a little bit more on...
Well, I guess...
in other words, these are happening together, but what causes what?
Right. So correlation is not the same. Yeah. But I guess one ambiguity you are revealing by fitting a model is this explaining away of parameters. Okay, so that's one thing I was showing you about how sort of couplings that you see measured. Just because you measure a correlation doesn't mean that there's something there, right? That's part of the causality question. So fitting a model goes part of the way in explaining away those false apparent connections of causality. But in terms of the temporal sort of structure, another thing that might help here in the oscillation model is having the phase structure. I don't know if Killian wants to comment more, but on that question.
So I think your answer was right. There's still a principal limitation of this kind of model that you don't attack. What you would have to do is you really would have to intervene and make experiments with, like, say, stimulation, for example, and then you can use the model to predict what the effect of the stimulation is, and you can double check in the experiment. But by just observing what's happening in the brain, I think you cannot get at the causality question. Which region is changing another region?
Last question.
So I was really intrigued that the sparse coding model gave you something that looked a lot like wavelets. And I'm wondering if you've sort of tested that against wavelets to see how well it predicts different behavior.
Wavelets in the images or wavelets in the neural activity?
In neural activity.
I see. Good question. No, we haven't fit a wavelet model. through neural activity. But that would be that'd be a sensible first start to try to start with that as a basis. And then
Well, I mean, it'd be particularly interesting to see if in sparse coding there's better than wavelets. Lots of people use wavelets right now, right?
Yeah. Well, I mean, there's one aspect of the wavelets that when they, just over time, but I think that the main thing that the algorithm is learning is the spatial structure. That you're not going to be able to guess a priori from a wavelet in terms of so how best to distribute things across space. That's something you don't have to sort of get from the data. But in terms of the bandpass, the temporal structure of these learned functions is fairly bandpass and reminiscent of a wavelet kind of type. So maybe you could start with something like wavelet with time and then just run the space part. But I guess that maybe the word I think what's important here is that again these because these people have been so skeptical about these oscillations that I think it's important to show that
actually switch right
you can learn these oscillations directly from the data without any a priori filtering. Right? You're not doing any
switching it,
you're not
putting
doing
it again.
very nominal bandpass filtering. It's just emerging straight from the data. Right? Whether you like it or not, it's saying, well, it's there. Right? It's being pulled out as a component. And that's part of why we're trying to do this learning approach as opposed to sort of just simply slapping it.
Oh, you actually put it in the two. Okay, that's the reason. Oh.
Specific neuroband frequencies that emerge from the data.
Sure. Okay. So thank you so much. Okay, Bruno.